Hello everybody, this is Dr. Fu. We're going to talk about optogenetics this week, and that's going to be our last module of the semester. Before we talk about optogenetics, we have some terminologies that we have to review in genetics. First one, DNA, chromatin, chromosome, and gene. Are they the same? The answer is no. They're very similar, but they are not the same. First one, the DNA, they are your genetic materials. Everybody know that, right? And then what is chromatin? Chromatin is the loose DNA. Like they lay in your nuclei the way that they want. They're pretty messy. And then the chromosome, they are neatly organized, the DNAs. So they lay in your DNA in a very organized format. So because of that, then you end up have 46 chromosomes in every single cell in your body except the sperm and eggs. Next one is gene. A gene is a piece of DNA that will code a piece of protein. So a gene is a more shorter piece of DNA. Okay, next terminologies. What is the transcript and translation? Transcript is basically you are making, or your cell is making RNA based on the DNA. So the cell making RNA based on the DNA, we call them transcript. And translation is the cell is making protein based on the RNA. So the RNA that we use is the messenger RNA, the mRNA, right? We cannot make DNA we cannot make protein directly from DNA. We have to make RNA first, and then we get protein out of that. Okay, what is gene expression mean? Gene expression means we are making a piece of protein based on piece of DNA. Okay, so we said the gene expressed, which means that cell making some protein based on that gene. Okay, here's a question. Every single cell of you have the exactly same genes. They have the same DNA, the same genetic materials, except the sperm and eggs. The question is, do they express the same protein? The answer is obviously no, because if they do, then you become a chunk of meat. But how does the cell with exactly the same genetic materials express different proteins? We're going to talk about that later in this lecture. Okay, here's the objectives. Particularly, we're going to talk about the Gulf-4 UAS system. It's a pretty complicated concept, and this word sounds very alien. How do I remember that? Okay, so I remember these 4 American girls. Okay, what is the thing? Gulf-4 UAS system. The UAS sounds like USA to me. Gulf is another terminology for girl, right? So there are 4 American girls. Gulf-4 UAS system. You have to remember them. Okay. And we're going to explain how they work. Okay, 4 American girls. All right, next one. We're going to talk about Drosophila. Okay, so Drosophila is a very powerful model for neuroscience research. Why is that? Because they are very small and easy to handle. Right? Have you ever seen Drosophila before? Yes, you do, if you're lazy enough. Because if you have your kitchen trash laying there for a week, you are going to have some Drosophila. They are the fruit flies. Okay, and they have very short generation time. They proliferate themselves in just a few days. So next, probably in a week, you are going to see the numbers of flies doubles. And then their genetics are easy to modify. That's why a lot of neuroscience research prefer Drosophila. Not only neuroscience, actually there are people study diabetes. They also use Drosophila as their models. Okay, before we talk about optogenetics, let's talk about some cool proteins. Without these proteins, the optogenetic wouldn't even exist. Okay, what are they? The first one is channel rhodopsin. The other one is holorhodopsin. Okay, so channel rhodopsin is light-gated, non-specific cation channel. So what does cation mean? That means the positively charged ions. So these channels will allow Na to enter the cell if they are open. So if they're light-gated on channel rhodopsins, they will be open. So that's what does light-gated mean. So if you have sodium go into the cell, the membrane potential is going to increase. And it's going to be easier for this cell to reach threshold to have action potential. So you will be able to activate these neurons. For holorhodopsin, they are light-gated ion pumps. They will typically pump chloride into the cell. If you pump chloride into the cell, because the chloride is negatively charged, you are going to decrease the membrane potential, right? So that's going to be harder for the cell to reach threshold to have an action potential. So these neurons will be turned off with light if they have holorhodopsins on their cell membrane. Other than holorhodopsin, channel rhodopsin, there's another type of rhodopsin called georhodopsin. This is also commonly used in optogenetics. This type of rhodopsin will pump hydrogen outside the cell. If you pump hydrogen outside the cell, you are going to decrease the membrane potential. If your membrane potential is decreased, it's going to be harder for the cell to reach action potential, so these neurons will be turned off. Okay, let's talk about the special channelrhodopsin chromosome. So what is special about chromosome is it can be activated by red light. And there's some other channelrhodopsin that responds to blue light. Now, what is special about the red light? The red light have a better tissue penetration than blue light. So that you can reach deeper, the red light can reach deeper than blue light. And they have less absorption by the blood. They also have less interfere with normal functions in Drosophila. So chromosome is being used in Drosophila study a lot. Alright, these proteins are pretty cool, right? However, neither human or fruit flies has any of these cool proteins, which means I cannot turn your neurons off with light even I don't like you. So, here's a question. If you want to have these cool proteins in our neurons, what do we need to do? First, we need to insert the genes for these proteins into our genomes. Then we have to do something to make sure these proteins only express in the neurons. Okay, let's talk about how to insert genes into your DNA. In order to do that, you have to go back when you are still embryo, which means you are still a few cells. If that's the case, then we'll be able to put a piece of DNA in every single cell of you. Embryo. How do you insert a piece of DNA into one cell? There is different gene transfection techniques, with CRISPR-9 is one of them. So CRISPR-9 is an enzyme-based method that you can insert any piece of DNA, pretty much every piece of DNA into a cell genome. So if you have that piece of DNA in your cells when you're embryo, and then when your cell divide, you are going to have this new piece of gene in every single cell. Then how do you control these new genes only expressing neuron? Okay, the answer is by controlling its promoter. Okay, let's talk about what is a promoter. Promoter is a piece of a DNA that will attract RNA polymerase. So for gene transcript to happen, which means from DNA to RNA to happen, you need to have RNA polymerase to bind to DNA first. And then the DNA region that polymerase start to bind is called the promoter. Okay, here's an example. This is a DNA, and then the RNA polymerase will bind here. It will start a transcript here. Okay, if you draw another illustration, this is the ATCG sequence of the DNA, right? And because the ATCG have 3D structures, the RNA polymerase also have 3D structures. They have to match. So they have to find a specific sequence on the genome to find their binding site to start the transcript. So this sequence, we call them promoter. Without this promoter, none of the downstream DNA will be transcribed into RNA. Okay, there are some special names of certain promoters. Some promoters are called enhancer or upstream activation sequences. Particularly the upstream activation sequences is something that we are interested. We call them UAS, the American Girls America, right? Okay, so they are basically just promoters. Okay, and some promoters will need ligand to activate. The ligand can be RNA, can be protein, or some inorganic chemicals such as zinc. Okay, here's an example. This is a promoter that don't require any ligand. This is a promoter that will require a ligand. You see there's a shorting here. If you have a ligand here, the bond between the DNA and the RNA polymerase will be very strong, so they will be able to start the RNA transcript. However, if you don't have this ligand, the bind between the DNA and RNA polymerase is going to be weak, so they won't be able to start. So if we control the presence of the ligand in certain type of cell, certain gene expression can be turned off and on. For example, if this promoter requires this ligand, if we don't supply the cell with this ligand, you are not going to be able to express the downstream gene after this promoter, right? Okay, this is how the epigenetics works. Let's say if your diet is constantly missing zinc, and then all the gene expression required zinc will not be able to happen. That way, your body will some function will change, right? That's how your diet will affect your gene expression. Okay, here's a question. GFP is a green fluorescent protein. It is coded by a gene called GFP. So usually when we talk a gene, that's lowercase, and that protein is uppercase. So if you have this protein in every single cell, you can turn green under blue light. Okay, so how do you turn yourself green? I will give you 3 pieces of synthetic DNA, which means these DNA are chemically synthetic. Okay, and you will be able to insert any of them into your genome. And if you want to turn green, which one you need to insert into your genome? Of course, it has to be before you were born, using CRISPR-9. Let's take a look. The first one is the universal promoter, which means this promoter will be activated in every single cell in any situation. And downstream of this promoter is going to be the GFP gene. And then next choice, I can give you a beta cell specific promoter, which means this promoter only activate in beta cell, and downstream is the GFP gene. And then the third choice is going to be a GFP gene only. So hopefully your answer will be the first one, right? Because without this promoter, the GFP gene will not express. You can have GFP gene in your genome. If your GFP gene don't have a promoter before it, it's not going to become a protein. And if you choose the second one, the B, the beta cell specific promoter, your beta cell will only have GFP, not other cells. So if you want to turn green, every single cell has to contain this GFP, right? So the answer will be A, universal promoter plus the GFP gene. Now let's talk about a special promoter, the UAS. That will bring us to this American Girl thing. That's a GAL4 UAS system you have to remember, okay? So let's take a look. For this UAS system, the UAS, like we talked earlier, it is a promoter. And this promoter requires a special ligand, which is GAL4. And GAL4 is a protein. All right, let's talk about some terminologies before we move to the next step. First one, in fruit flies, different neurons will report different signals to the brain. So the neuron detected sweet will not be activated when there's a bitter taste. So Dr. Fu called these neurons sweet neurons. And then same thing for bitter neuron. By saying bitter neuron, I mean the neurons will detect a bitter taste. Okay, so the sweet neuron and bitter neuron. And then the sweet neuron specific promoter. So these are the promoters can only be activated in the neuron that detect sweet. And then the reason why they're sweet neuron specific promoters is because the ligand needed for this promoter only exists in sweet neurons. So even bitter neurons, they may also have this promoter. They have the sequence in their DNA, but it won't be activated. Why is that? Because they don't have the ligand. Remember, this ligand only exists in the sweet neuron. Okay, so with that being said, I have another question. Let's say if you have a fruit fly that has 2 pieces of DNA that inserted into its genomes. Okay, so the first piece of DNA is a sweet neuron specific promoter plus a GAL4 gene. Okay, what is a GAL4 gene? The GAL4 gene is the DNA that will code for a protein called GAL4. Do you remember GAL4 earlier? That is a ligand for our UAS promoter. Is that right? Okay, so that's the first piece of gene, the promoter plus the gene. Okay, and then the second piece is the UAS, which is a promoter plus a chromosome gene. Okay, if you remember, chromosome is a special channel rhodopsin, right? So now here's a question. In which cell will chromosome will be expressed and why? Suppose a fruit fly has every single cell have these two pieces of genes and asking you which cell the chromosome will be expressed and why. And another question is, will you have chromosome protein if you don't have a DNA piece A? If you don't have this, you only have this. Are you going to still have chromosome protein? Okay, here's the some clues. First one, UAS is a promoter, and that will need ligand a GAL4 to activate. We talked that earlier, right? And to activate a promoter, that means the downstream gene expressed, which means you're going to have some protein out of that. And GAL4 is a DNA sequence, and GAL4, the capital, is actually the protein that being made using this GAL4 gene. Okay, and then the chromosome is a DNA sequence that can make a protein called chromosome. Okay, so there's two pieces of genes in the fruit fly. Which cell you are going to have chromosome expressed? The answer is in sweet neuron. Why is that? Because only in sweet neuron you have this promoter. This promoter will drive this gene, which means this gene will be expressed, right? In sweet neuron, you are going to have GAL4 protein, and once you have this GAL4 protein, the GAL4 protein will bind to this UAS promoter, and once the GAL4 bind to UAS promoter, it's going to be activated, which means the downstream gene, which is chromosome, will be expressed. So in this sweet neuron, you are going to have chromosome protein, right? And if you don't have this piece of gene, you are going to have UAS promoter drive a chromosome gene, but this UAS is not going to be activated because it's lacking its ligand, the GAL4, right? So the answer for this one is no, you don't have it because you are missing a ligand for your promoter. All right. Okay, so here is the structure of that gene. If you have a sweet promoter here, drive a GAL4 gene, and these whole things has to be in one cell, right? In together is within a cell, because GAL4 is pretty big. If you have this one in separate cells, this one in one cell, this one in another cell, the GAL4 won't be able to migrate to another cell to reach the UAS to activate that. So everything has to be in one single cell. So once this gene is activated, you are going to have GAL4 gene, and you are going to have GAL4 protein. The GAL4 protein will bind the UAS, and this will be activated, and then you're going to have your channel rhodopsin or chromorhodopsin expressed. So here is another question. I have five flies with different genetics background. The first one have a UAS promoter plus a chromosome gene. The second one is a UAS plus holorhodopsin gene. Okay, and then the sweet neuron promoter plus GAL4 gene, beta neuron promoter plus GAL4 gene, and beta cell promoter plus GAL4 gene. By the way, the beta cell is the cell in your body which make insulin for you. Okay, so here's the question. Which flies do you need to mate to get their baby? So the baby, first one, you can get flies that can turn on sweet neurons by light. And then the second flies, you want to turn on the beta neuron by light. And then the flies that you want to turn off the beta cell by light. Okay, so which flies do you need to choose as a parent? So the baby flies you can get will have these features. Okay, let's take a look at the first one. If you want to have flies, they can turn on neurons by light. If you turn on neurons, you would choose chromosome, right? Because this one will turn on neurons. The holorhodopsin will turn off neurons. So obviously you need this gene in there. And then this flies carries this gene, and this one has a promoter. In order to activate this promoter, you need a ligand, right? Which ligand? You need GAL4, and all of them have GAL4. And in this particular case, you need to turn on sweet neuron. So you want a sweet neuron specific promoter, drive GAL4 gene. So this gene will express only in sweet neuron. And in your sweet neuron, you have this GAL4 protein will activate UAS promoter will drive chromosome gene, which means you're going to have chromosome protein. Okay, so the first answer is going to be fly A plus fly C. So their baby is going to have their sweet neurons can be turned on by light. And for the second question, you want to turn on the beta neuron. So the same thing, you will choose fly A as one of the parents, and then you need a beta neuron promoter drive this GAL4 gene, which is D. So A and D for the second question. And then if you want to turn off beta cell by light, you are going to use holorhodopsin, which is this guy, carry a holorhodopsin, and you need a beta cell promoter for E, right? Okay. Let's talk about channel rhodopsin. Channel rhodopsin will open under light, so you can control the function of a nerve. However, they only open under the light when there is a retinal or vitamin A. If we don't supply vitamin A or retinol, the channel rhodopsin will not open even there is light. So we have to make sure that we supply retinol or vitamin A to the fly food. Same thing for chromosome, which is a special kind of channel rhodopsin, right? We also need to supply vitamin A in the fly food. Let's talk about fly lines. So fly lines is not a line of fly. They are a group of flies that share the same genetic background. So in this lab, we're gonna have two fly lines, which means we're gonna have two group flies. Each of them contain the same gene background. So for these two fly lines, each of them contain two pieces of exogenous DNA, which means these two DNAs are inserted into their genome. Okay, for fly 1, we have two pieces of DNA. One is sweet neuron specific promoter drive a GAL4 gene. So this piece of DNA will make sure this GAL4 protein only expressed in sweet neuron, right? And then in the same fly line, they have UAS crimson gene. So because this fly have GAL4 in their sweet neurons, so in their sweet neuron, their UAS will be activated, their chromosome will be activated. So this fly line, they will have sweet neuron express chromosome. If you shine a light on them, their sweet neuron will be activated, right? And another fly is beta-specific neuron promoter plus GAL4 gene, and then you have UAS plus chromosome gene in that fly line too. So for this, the second fly line, the GAL4 only expressed in beta neuron, and in beta neuron, only in beta neuron, the UAS will be activated, which means the chromosome will be expressed in the beta neuron, right? Okay, so we need to do an experiment to genotype these two fly lines. So what does genotype mean? Genotype means predicts their genetic background based on their behaviors. Okay, so what is the expected result we're gonna have? Experiment that we do here is based on the assumption fly will like sweet and hate bitter. So if we provide them sweet, they would like to go there. If we provide bitter, they don't like to go there. So for the first fly line, which contain these genes, if you shine a light on them, a red light on them, do you expect them avoid or prefer red light? The answer is they will prefer red light, right? Because their chromosome is expressed in sweet neuron. So if you shine a light on them, their chromosome will be activated. That means they are going to activate their sweet neuron. So they think they have sweet food in there. And then for the other fly line, if you shine red light on them, they probably will not like it because they think that red light is bitter, right? So before we do this experiment, we actually need to fast the flies overnight before we test them. Why is that? Because if the fly already well fed, they probably don't care sweet thing that much. So if you fast them, that will enhance our result. Okay, what is special about the fruit flies is they have taste nerve that exposed to the environment. So this is their mouth. You can see all these antennas here, all these small spikes here. They have nerves exposed down here. So the light will very easy to hit on their nerves. We don't have to penetrate deep into their mouth to reach their taste nerves. We're going to use fly maze to test their light preference of our flies. So this is a 3D printed fly maze. Commercially, there are different type of materials they make fly maze, but they basically use the same mechanism. Okay, so this is the fly maze. How to set up the fly maze? There is a piece of a hole in the centerpiece. They're connected. So if you turn on the light here, the flies will either stay under the light or go in the dark. So that way you can test if the fly like the light or they don't like the light. And something special is in the centerpiece here. There's a hole in the centerpiece. You'll see that later. the flies, if they stay in the centerpiece, which means they probably don't like light either because the centerpiece is dark. So when you're calculating the preference index, you are going to use the numbers flies that you got in the light, which is in this tube, and then you minus the numbers in the dark, which is in this tube, and then you also need to minus the number of flies in the middle part, the centerpiece, because that's considered as dark too. And then you are going to divide it the total fly numbers. That's how you calculate the preference index.